So I'm Jason Ernst. I'm faculty here at UCLA, and I'm going to talk about computational approaches for deciphering the non-coding human genome. So this is a small portion of the human genome, and one key aspect of this is only about 1% of it codes for proteins, the relatively well understood parts, and 99% about is non-protein coding. And this area of the genomes of key interest has been highlighted by genome-wide association studies, which you heard about yesterday during Melissa's talk. And one key aspect of this is in these unbiased mapping of where disease-associated genetic variation is a large majority of it is falling in these non-protein coding portions of the genome, and then many are assumed to have regulatory effects. So there's a need to better understand this non-protein coding portions of the genome. And what I'm going to survey in this talk are 3 different strategies to interrogating these parts of the genome and talk about some computational methods that I've been involved in developing in each of these areas. So the 3 areas I'm going to talk about are: 1 is using epigenomic signatures, 2 is using high-throughput functional testing data, and 3, comparative genomics information. So if one tries to read the DNA sequence directly, it's very difficult. But if there was post-it notes that marked important parts of the genome, this could give us a clue on where to sort of focus our attention on. And there might be certain patterns in these. You could think of markers on the genome in terms of we might see various markers of different types reoccurring together. So this could be a signal that there's something important underlying these locations, that we would see these markers co-occurring together repeatedly. And we might see broad domains of other markers. And this could be another signal. And biologically, what we have this in is epigenetic marks. In particular, we're going to focus on histone modifications. So these are chemical modifications to the histone proteins that the DNA is wrapped around. So the DNA is wrapped around nucleosomes, which are an octamer, 8 histone proteins. And these histone proteins, and often the tails of them, can be subject to a variety of chemical modifications. And there's specificity in terms of which specific histone protein's modified, which specific amino acid residue of the histone is modified, what type of chemical modification is, whether it's a methylation or acetylation, and in how many occurrences it's occurring. It could be 1, 2, or 3. And then there's sort of a nomenclature that you might see around. For example, this H3K4ME3 means the 3rd histone, the 4th lysine, and 3 copies of methylation. And what has generated a lot of excitement in the field is that we now have the ability to map these histone modifications genome-wide based on this technology called ChIP-seq, chromatin immunoprecipitation followed by high-throughput sequencing. And in a typical experiment, one can get on the order of 20 million reads across the 3 billion bases of the genome, giving you density maps of different histone modifications where they are across the genome. And you don't just have one of these histone modifications, you can have potentially dozens of them mapped in a single cell type. So I've worked on this question: given that we had all these various maps of these histone modifications, could we de novo discover what are the major reoccurring combinatorial and spatial patterns in this data? And then once we've identified and defined these patterns, could we go across the genome and annotate each location as being an instance of one of these patterns? And this would give us an annotation of not just the coding portions of genes, but the entire genome, including the introns and intergenic regions. So computationally, what we used was a multivariate hidden Markov model, which would both capture the combinatorial nature of the data and also the spatial information. So what we did for the hidden Markov models, we had a product of independent Bernoulli random variables as our emission distribution. And we pre-processed the data to have 200 base pair non-overlapping intervals. And we used a Poisson background distribution to call if a mark was present or absent within each 200 base pair interval. And then what we would do is we would learn the parameters of these binomial the Bernoulli random variables de novo based on the data, and then there would also be transition probabilities which could allow us to capture spatial information. For example, this location, we might not have seen submark in a specific 200 base pair interval, but based on the neighborhood information, we could still infer we're in this state 5 associated with one histone modification, while here we might be in a state not really associated with any modification with high probability. So we applied this modeling framework to a set of CD4T data mapped by Keiji Zhao's lab based on 41 chromatin marks. And what you're seeing here are the rows correspond to the different chromatin states, the columns correspond to the different marks, and then the value corresponds to what's the probability we would observe that mark in that state. And to give an example of one of these states here, what I'm showing you is this enrichment for zinc finger genes and CAP1 binding, and the blue means a higher enrichment. In this case, there's over 100-fold enrichment for a class of zinc finger genes, specifically in this state here, which was associated with 3 of the histone modifications, H3K36ME3, H3K9ME3, and H4K20ME3. And it turned out that a large portion of this abstract of this paper was describing specifically that the presence of CAP1 and zinc finger genes were associated with these 3 histone modifications and the lack of a certain other marks here. And then we more systematically showed the lack of all these other marks. So this is an example where by just applying the right appropriate computational machinery to this high throughput biological data, we could discover a very specific biological concept de novo. And we more systematically assigned every location in the genome to one of these states, and then we computed enrichments for the other categories that weren't used in generating the model in terms of annotated genes, evolutionarily conserved elements, other experimental data, and gave a biological interpretation to each of these states. And what we characterized was different classes of promoter states, different classes of transcribed and intergenic regions that were active, and particularly important are these class of elements called enhancers, which can be potentially far away from the genes, but important in turning genes on and off. And these can be difficult to identify without this type of information, and also various types of large-scale repressive and repetitive elements in the genome. So that was all in one cell type, but one of the real powers of the epigenomic data compared to just looking at the DNA sequence is you can gain access to cell type specific information. So the genome is pretty much the same in every cell type of your body, but then if you look in different cell types, the epigenome could be very variable. So you could see in different locations, different marks move around. And so in collaboration with Brad Bernstein's ENCODE production group, they had mapped a subset of the marks in a variety of human cell types, 9 at the time. And this is looking at the data in one genomic location. So we have a variety of different histone modification maps. The colors correspond to different cell types. And what we did was we applied the same modeling approach, but first conceptually concatenated different cell types, treating them as if they were different chromosomes. What we would learn is one common set of state definitions across all the cell types, but cell types specific state assignments. And it turns out that the combinations of marks that you would see across different cell types tends to be relatively constant across cell types, but locations of them can move around. And we did a similar thing. Once we had learned one of these models, then we compared each state to a variety of other external annotations and data, and gave candidate annotations to each of these states, whether it was associated with like an active promoter or more of a poised or inactive promoter, various classes of enhancers, insulator, transcribed, repressed, heterochromatic, and repetitive elements. So this is another view of what we've done. You're looking at one genomic location in 4 different cell types. These rows correspond to the heat map density of the various modifications in each of the cell types. And then this row on bottom is the chromatin state call for that location and that cell type based on this color coding. And then one can now take some location they're interested in the browser and then quickly scan across any location and see its chromatin state assignment in each of those cell types. So then once we had these genome-wide annotations in a variety of cell types, we started revisiting some of these GWAS hits that were coming out of the genome-wide association studies. And it was sort of, at the time, an issue that a lot of these hits were coming in these non-coding parts of the region without other types of biological support of their importance. And this is giving you an example of one of the studies we sort of revisited at the time. It was looking at erythrocyte phenotypes, which can be associated with hypertension and cardiovascular disease. And what you see here on the rows correspond to the different cell types we were considering, and then the columns correspond to different lead SNPs from this GWAS. And what you'll notice here is that there was a large block of SNPs that were being assigned to what we were characterizing as strong enhancer states. And if you're familiar with the biology of each of these cell types in this phenotype, this is specifically the biologically relevant cell type to this phenotype. And we more systematically went through the GWASs that were available in the catalog, GWAS catalog at the time. And what we found was there was a number of GWASs where by just taking the top few SNPs, the lead SNPs that were curated into the catalog, and comparing them to our calls of these strong enhancer states, we could find enrichment for these SNPs in these strong enhancer states, and particularly in the biologically relevant cell types to the phenotype, in terms of we saw a lymphoblastoid associated with some immune-related, HepG2, which is a liver-related cell type with cholesterol and lipids, and then K562 with erythrocyte and phenotypes. And others have now sort of more generally used this type of data in additionally more sophisticated ways and have seen similar types of relationships. And since then, these chromatin states have had a wide range of applications. By many in the community, I'm highlighting some that collaborations that I've been involved with and some people in the room have, where we've used these chromatin states to look at iPS reprogramming, melanoma progression, and chromatin interactions of the developing brain. And also I collaborated with the Roadmap Epigenomics Consortium, which had this goal of creating a set of reference epigenomes in a large number of different primary cell and tissue types, which was differentiated from the ENCODE project at the time, which had been primarily focused more on cell lines. So what you're seeing here is a set of various epigenetic marks that the project was mapping, mostly histone modifications. Another important one was DNase I hypersensitivity, which measures open chromatin or accessible DNA across a wide range of the human body. And one of the sort of key aspects of this data was that there was 5 core modifications that were mapped in almost every cell and tissue type, and then a couple other primary marks that had fairly deep coverage, and then it was sort of sparser for a number of other marks. So the rows correspond to the different epigenomes and grouped by their sort of major classes of cell types here. So what we did in the project was we learned a chromatin state model based on 5 histone modifications, which were mapped in every one of these epigenomes. And so it's defined across 127 cell and tissue types based on these 5 marks. And then we wanted to have another model which included another important histone modification, H3K27 acetylation, which can be associated with active elements. But then that one wasn't mapped in every single cell and tissue type, so we only defined it on 98 of them here. But then this sort of raised this more general question. I mean, we could keep on adding marks and reducing the number of cell types, but could we have an alternative strategy? And what I developed was a method which could computationally impute some of these experiments. So we heard about imputation from Donna Pierre's talk. In this case, we're trying to impute an entire experiment, not just single missing data values. So what we're trying to do is effectively fill in one of these experiments. How do we predict this mark in this cell type if we have any data for that? And what we're going to do is we're going to leverage the fact that we've seen that mark in other cell and tissue types, and we've seen other marks in that cell and tissue type of interest. So we can use both classes of information, leveraging that Mark's correlation structure can be relatively well conserved across cell types. And we can also look up that mark that we're interested in at that specific position, what it looked like in closely related cell types. So we defined sort of features that capture that information, and then the framework we used was an ensemble of regression trees, which Jennifer Listgarten explained the regression trees in her talk. And then it turned out that, so we not only predicted some of these mark experiment combinations that we didn't have any data for, but we also generated an imputed version of all the data sets for which we did actually have data for, not using that specific experiment imputation. It turned out there was a lot of nice properties of the experimentally imputed data, even compared to the observed data, because there was a statistical robustness we gained when we used information from dozens of very closely related and informative experiments, compared to if you rely on the data from a single experiment. There can be a lot of confounders in terms of experimental variability, the genetic background, and environmental effects that can be sort of averaged away through the imputation process. So this is just giving you a visualization of the performance here. So the blue is the observed data, and red is computationally imputed data, not using that observed data in the prediction. And I'm showing a different cell type for each mark here. And now we're zooming in, and we did this up until 25 base pair resolution, except for DNA methylation, which we did at single nucleotide resolution. And we also did more quantitative evaluation. So for each mark, we compared it to sort of two stringent baselines. One is how well it would do against if we took all the other experiments for that mark and just averaged them together. And then the other baseline was what's the best case if we took an experiment for that mark we're trying to predict and just use that in place of that experiment. And in either case, our imputation did better than averaging, or the best case, no matter how well we could choose some other experiment to use in place of it. So once we had this more complete matrix, we then defined a set of chromatin states, in this case, based on the 12 marks we were most confident in the imputation, based on having enough other cell and tissue types where that mark was experimentally mapped, and then defined a chromatin state model with 25 states across all these various cell and tissue types. So this is a screenshot at one location. So it provides a very deep and rich annotation of the human genome into these chromatin states. So now, next part of the talk, I'm going to talk about some work on using high-throughput functional testing. So this is trying to get at sort of two limitations of this epigenomic data. One is that it's only sort of these biochemical signatures, and these are just like candidate enhancers often we're describing. It's not giving you direct functional testing data that this is actually doing something at specific locations. And then the other thing is we'd like to also start drilling down closer to more the nucleotide resolution. Often epigenomic data can be at a sort of 200 to 300 base pair resolution. So what we wanted to do was test thousands of these regulatory regions in parallel and identify the activating and repressive sites and distinguish activating from repressive. And in this case, we wanted to do this independent of the DNA sequence. So there's other work, some of which James mentioned, where people are trying to take models of the DNA sequence. But here we wanted sort of more direct experimental evidence that some site is active or not requiring on sequence modeling. So the enabling technology for this work is something called a massively parallel reporter assay. The specific technology that I worked with was developed by my colleague Charjay Mickelson while we were both at the Broad Institute, and a number of other groups have developed related technology. So how this works is that one can synthesize tens of thousands of DNA sequences that they're interested in testing, and then those sequences can be placed in upstream of a reporter gene, and then there's a unique barcode to each sequence that can be placed in the 3' UTR. So if that sequence you design is able to activate the gene, then what will happen is that barcode will be expressed. So then you sequence the barcode and you get a count of how often that sequence you designed is able to drive gene expression. So this gives you a quantitative readout of your design sequence's ability to drive gene expression. So the first application of the technology that Tarjay developed, what they did was they took two well-known enhancers and they did a systematic mutagenesis. They mutated each nucleotide in each possible way, and they did multiple different barcodes to help control for noise in the assay. And this gave a very detailed view of those two enhancers of where the activating and repressive sites were. But because it was devoting so many different barcodes and sequences to interrogating just these each location, they could only test two enhancers overall. In a follow-up work led by my colleague Puya Kradpour, what we did was we took more than 2,000 of the enhancers that we had predicted based on the chromatin states that also overlapped a regulatory motif. So these are patterns in the DNA which specific transcription factors bind, and we took these motifs that we predicted were important in these cell types, and we also did targeted mutations to those motifs. And this was largely a confirmatory study. It both validated a large number of these enhancers were active in the cell type, and also that the mutations that we thought would impact regulation often did. But we were interested in, could we effectively combine the elements of these two different studies? One is test thousands of locations in parallel, and two, to identify at a very high resolution the activating and repressive sites de novo independent of DNA sequence. And the challenge here is that there's this constraint on the number of sequences you can interrogate in the assay. It was originally in the tens of thousands. It's now gotten up to about 200,000. So the challenge is the more sequences you devote to interrogating any one location, the fewer locations overall you can test. So we ended up converging on a fairly simple, but turns out to be powerful design. What we would do is we would take some location we're interested in testing, and we would tile it with 31 reporter sequences. Each sequence is 145 base pairs in length. That's sort of a limitation of the DNA synthesis process. And then we put them in 5 base pair increments, and we centered them on a regulatory site of interest. And this had a number of advantages. One is, in a single experiment, we can interrogate close to 8,000 regulatory locations. And then also we have robustness to not knowing the specific 145 base pairs to test because of, from the DNA hypersensitivity or other chromatin data, we can often be confident at 200 to 300 base pair resolution, but it's hard to be confident of the specific 145 base pairs. And then we get robustness to noise based on the high density of the overlaps, but that's also what gives us access to the high resolution information by carefully reasoning about the relative activity of these different tiles. So in collaboration with Tarjay's group, which did the experimental work, what we did was we tested more than 15,000 locations. They were all centered on a peak of this open chromatin DNase I data, and then we did a stratified random sampling to interrogate different classes of chromatin states from these models. So one of the goals was to understand the differences of the states. And we ended up testing in 2 different cell types with 2 different types of promoters, 2 sets of sequences based on the same design strategy, but they were different sequences, and then everything was done in replicate. So this is giving you some intuition about how one can go about thinking about this type of data. So if this was 295 nucleotides of sequence, and these are the 145 base pair subsequences, and gray represents basal expression. If there was an important activating sequence sitting right here, we'd expect these subset of tiles to be higher. If there was an important activating sequence sitting right here, we would expect these tiles to be higher. And if there was an important repressive site right here, we would expect these to be below the basal expression level. So that was just a sort of a toy example. Here are 2 examples of real data. And what we need to do is take data like this and come up with some probabilistic score of the relative activity of each position in the region. So this is showing you here, in one site, we're making a prediction that there's an important activating site right here, and here we're making a prediction that there's some important repressive site right here. Black is our overall prediction, and green and blue are based on effectively 2 different replicates. And then we can compare this to other type of information. So and what you see in red is a prediction based on the centipede method from Jonathan Pritchard's group, which predicts regulatory sites based on primarily DNA sequence motif information. And it can't differentiate activation from repression, but you can see it aligns very nicely with our predictions here. So we both add the activation and repression information, and our predictions are independent of any DNA sequence information. So we could potentially identify activating and repressive sites that might not closely match with a certain DNA motif. So to give a little more details about how we go from that data to these scores of the relative activity or of repression or activation, what we do is we set up a probabilistic model here where the Y's denote the observed variables, the H's denote the hidden variables, and we have a hidden variable for every 5 base pair intervals. So we had 295 base pairs in total, so we have 59 5 base pair intervals, and we have up to 31 observations. And then we make certain modeling assumptions. We assume that the Y's are normally distributed with their mean set to the mean of the H's that it overlaps, so that there's an edge if that hidden variable overlaps the tile that the Y corresponds to. And then the variance of the Y's are set to the empirical variance of the data, and then we put a normal prior on the H's with their mean set to the empirical mean of the observed data, and then we have one free parameter here for the variance prior there. And in practice, what we do is we set it to a loose and tight setting, and then at each position we take the more conservative value after standardization. So this is showing you more examples of our predictions. In this case, I'm showing you every location that we tested that had this motif predicted by Centipede to be active in the cell type in red, blue or other motifs predicted by Centipede, and then black is our inferred activity score across the 295 base pairs. And you can see that it's almost always above average. Sometimes it peaks over that motif. And now this especially contrasts if we take another motif. Now you're looking at all the sites we tested that had an NRSF motif predicted in this to be active in the cell type. And you can see that it's almost always below average, and quite often there's a strong dip in activity right where that motif is sitting. And again, we've made these predictions independent of any DNA sequence information. And then we've done more systematic quantitative evaluation, showing as we go to higher activating and also more repressive scores, we have a greater fraction of overlap with these annotated motif predictions from the Centipede method. And then we can also go into individual motifs and compute what's their average activity score, and we can see, for example, REST was a consistent repressor in both cell types. We had some motifs were a consistent activator. An interesting finding that came out of this work was the motif that had the strongest activation in both cell types was the tenmer, which doesn't have a sort of agreed upon protein that's associated with regulating it, even though it's come up in a number of other contexts as being one of the most conserved motifs evolutionarily, and also associated with some of the highest expression in vivo as well. And then if we look off diagonal, we can see some cell type specific activators, and also in some cases cell type specific repressors. And then that was looking at the average activity of the motifs. We're also powered to look at whether there's some motifs that are working as both an activator or a repressor in a given cell type. And what we found were most motifs were either primarily an activator or a repressor, so we're testing enrichment above a certain activation or repression threshold, but we did see some examples of motifs that did show some activity, particularly in the MAF family. And then we could also look at repetitive elements. We found ERV1 repetitive elements were actually some of the most highly activating sequences that we tested in this assay, while there's other repetitive elements such as line elements which showed neither activation or repression. And then the last thing here was we then compared our original chromatin state predictions in terms of the relative activity. In this case, we're taking all those locations that were selected based on being a DNase peak in HepG2 and showing their average activity in each of the chromatin states. And we found what we characterized as an active promoter and active enhancer had above average activity. Even though all these locations were an open accessible region, the additional information from our chromatin state calls could differentiate the relative activity here. Now, the final part here, this other strategy for annotating the genome I'm going to talk about is using comparative genomics information, and this is some of the newest work that's currently unpublished. So the comparative genomics data is sort of very complementary to some of the epigenomic data because it gives you an annotation of the genome even if you don't happen to have the experiment done in the relevant cell type to some location. Because if there's some regulatory element in the genome, and you didn't do an experiment in the sort of cell type where it's active, you might not see any signal relevant to it. But you don't have that concern with the comparative genomics data. And another nice aspect of it is that you get the single nucleotide resolution, which we're particularly more powered now that we have large numbers of species in these alignments. So UCSC has a 100-way alignment available. And traditionally, the representation of this type of data in a genome-wide manner has been based on either univariate scores or binary element calls of evolution constrained in terms of methods such as PhyloP, FastCons, SciPhi Omega, Pi, GERP, GERP++. And now these scores have been further become key aspects of other integrative scores that you might have heard of, such as CAD, GWAVA, Eigen, LNCite, and so forth. But there's some limitations of this that we're noticing, and sort of the issue is that we're trying to compress a lot of information about 100 different species into a single score or binary 1-0. So just taking, for example, 2 locations in the browser, you can see here these are the PhyloP and FastCons tracks. They're both showing a lot of activity, but the alignment is, in this case, I mean, it's an extreme example, but it's quite different. Here, you have all these distal species, distal vertebrates aligning without any closer mammals. So this is suggestive of some type of alignment artifact. While here we have sort of a more robust alignment. In some locations we might have alignment through all the vertebrates, but other times it might be through all the mammals or some subset of mammals. And that is potentially very informative information about the genome that's being lost if we're trying to compress everything into a single binary call or score. So inspired by the Chrome-HMM modeling approach, we were thinking, could we do something similar with this multi-species alignment algorithm? And we've developed this method which we call cons-HMM. And what this does is it tries to learn a set of conservation states based on the combinatorial patterns of which species align and match the human reference genome. So what it does is it takes a 100-way alignment or any alignment, but we worked with the 100-way alignment here, and we encode it as one of 3 possible observations. A species for each species. It either aligns and matches, it doesn't align, or has an indel there, or it aligns but it doesn't match, have the same nucleotide as the human reference genome. And then what we do as our emission distribution is we have a set of independent by multinomial random variables for these 3 observations. And we also do everything at a single nucleotide resolution, which compared to Chrome-HMM, which had worked on 200 base pair resolution, raised some scalability issues that we managed to get around. So this is another view of what we're modeling here. So this is the human DNA, and then these are the various species. And then what we have here are, for each state, we have different probabilities of each of these observations for each species. And now what you're seeing here is the output of our model in terms of the mission parameters. The right side is the matching probability, and here I'm representing the aligning probability, which is 1 minus the not aligning observation. And the columns are ordered in terms of distance to human or divergence from human. And then the rows correspond to individual states, which we've clustered and ordered computationally. And then, so red means a higher probability of here aligning and here matching. So we have, for example, a state here which is aligning through all the vertebrates, a set which has some coverage in some of the vertebrates, but not necessarily all. Here's where we have fairly good alignment and matching through most of the mammals. Here where we can be missing key subsets of mammals such as mouse and rat. While here we have aligning through most mammals but not matching. Here we don't even have aligning through most mammals. Here we're not even aligning through all the primates. And then this is an interesting one in that we have all these distal vertebrates aligning but not these intermediate mammals, which can be associated with potential alignment artifacts. And this is showing you the annotation at 2 locations, and this is the underlying alignment patterns, which even though they have similar constraint scores can have very different conservation state assignments. And then showing you that in terms of, for example, fastcons, we can have a large number of these elements enriching in these states, but also the state down here as well. And then we can compare these conservation state alignments to other types or calls, other types of genomic annotations. And for example, we were able to find one state which had more than a 30-fold enrichment around transcription start sites, and a different state, different states with more than 50 and 100-fold around transcription start sites of pseudogenes. Here it's of protein coding genes, just based on these combinatorial patterns of aligning and matching the human reference genome. And then this is an illustration of how we're able to get some of the single nucleotide resolution information. I'm showing you the enrichment patterns relative to the start of exons and the end of exons. And you can see the 3 nucleotide oscillation within exons, and then some of the splice site signals flanking it as well. And then these are the different states here. So for example, 3 and 54, which have this inverse oscillation pattern, have a deeper alignment but not high matching probabilities, while 1 has this sort of alignment and matching through all the vertebrates. And we can also compare these conservation state enrichments to the chromatin states. And what I'm showing you here are the subset of conservation states which were maximally enriched in terms of, for at least one chromatin state based on their median enrichment across 127 cell and tissue types. And we can get some conservation states which are strongly enriched for what we were characterizing as promoter states. This one was most enriched for what we were characterizing as a more active enhancer states, or transcribed states which have the exons and other patterns for other states. And then we can also compare how our enrichments look like if we condition on being in a fastcons element versus not in different conservation states. So what we did was we took the fetal brain DNase hypersensitive sites. So these are open chromatin regions in fetal brain. And then we can look specifically at the non-exonic regions. And we asked, what's the enrichment in fastcons in general? And it's about 7-fold. But now if we condition on which conservation state we're in, and we stay looking at a fastcons element, that enrichment can go anywhere from 17-fold to more than a 2-fold depletion. And I've ordered these states based on their enrichment for fastcons, for fetal brain within fastcons elements. And 20% of all non-exonic fastcons elements fall in one of these states or below. And in blue is enrichment not in a fastcons element, but in one of these states. And sometimes we even get higher enrichment than in fastcons elements. But this is also saying sort of complementary information from sort of this proper phylogenetic modeling and the conservation state assignments, both in a fastcons element, depending on what conservation state we're in, we can see a wide range of enrichments, but also if we're in a fastcons element in any given conservation state, we also see greater enrichment. And we, as the last two analyses here, we also compared our enrichments with locations of human genetic variation, and we saw more than 8-fold enrichment that's in red for certain conservation states for human genetic variation, and they were associated with a pattern of aligning at least through all the primates, but not having strong matching probabilities through most of the primates. And then if we compare that relative to the GWAS catalog, we didn't see that same enrichment. While on the other hand, the state here was most depleted for human genetic variation, but then the enrichment of the GWAS catalog relative to this state, it made it sort of the most enriched. And then the last analysis here, we applied the heritability partitioning method developed in Alcus Price and Ben Neal's group, where we took, so they had previously reported a large enrichment for constrained elements for a variety of enrichments for different phenotypes in constrained elements. But what we asked here is if we partition our constrained elements in two sets, sort of that are of equal size, where one's sort of considered a more confident set and the other a less confident set, where we define the more confident set as those conservation states that in general enriched for DNA sites. So we took the top 7 states in terms of enriching for DNA sites, and that contained 51% of, or 52% of all the FastCons bases, and then 48% were in the other 93 states. And then if we applied their heritability partitioning approach, we saw very strong enrichment for most phenotypes only in the FastCons that were in the top 7 states, but not in the other 93 states. So just to summarize, I presented to you sort of 3 strategies for annotating the non-coding parts of the genome, one leveraging epigenomic signatures and chromatin state annotations, another based on high-throughput functional testing. The method I showed you was using a massively parallel reporter assay tiling, and then a third approach was using this complementary conservation state annotations. And this work has been done with a number of collaborators, and then to highlight Adriana, who did the conservation state work. So thank you for your attention.